Okay, so in this video, I'm going to talk to you about secondary, tertiary, and quaternary protein structure. You should already have learnt about the structure of amino acids, the formation of peptide bonds, and the primary structure of amino acids. So just to recap that, we know that the primary protein structure is the sequence of a chain of amino acids in a single polypeptide, all joined by peptide bonds. So at the primary structure, the only bond present is peptide bonds. Once the peptide bonds have been formed during protein synthesis, the protein will spontaneously undergo a series of changes to firstly form hydrogen bonds and form its secondary structure, and then form further bonds to form its tertiary structure. Proteins that can then undergo further bonding to join two or more polypeptide chains together. And once this occurs, we call this the quaternary structure. So we are going to look at each of these structures in turn. So let's look at secondary structure. When a protein goes from primary to secondary structure, hydrogen bonds are formed. The formation of these hydrogen bonds can result in two distinct shapes, the alpha helix and the beta pleated sheet. The hydrogen bonds form between the NH groups and the C double bond O groups on two adjacent amino acids. So let's just take a look at that. Another shape that can form due to hydrogen bonding at the secondary structure is the beta pleated sheet. The beta pleated sheet has a wave-like structure, and the way that the hydrogen bonds are formed allows for more stretching of the polypeptide chain. An example of a protein with a beta pleated sheet would be the protein in feathers. So we've looked at primary structure and secondary structure. Let's now look at tertiary structure. When the tertiary structure forms, the protein chain spontaneously folds to form a three-dimensional structure, which contains further bonds. So in addition to the peptide bonds and the hydrogen bonds that we already have, we now also add disulfide bridges, which are made between the sulfur atoms found in the variable regions of the cysteine amino acids, ionic bonds, and hydrophobic interactions. And we're going to look at each of these in turn. So let's go through each one. Let's start with the hydrogen bond. So these hydrogen bonds are just like the hydrogen bonds found in the secondary structure. So whenever slightly positively charged groups are found close to slightly negatively charged groups, hydrogen bonds can form. What I'd like to do right now is I'd just like you to imagine this long polypeptide chain, and it's already got a little bit of hydrogen bonding. That was in the secondary structure. But the purpose of all these further bonds is to create this three-dimensional shape. And it's really important that you're able to visualize that in your mind. So it's not a flat two-dimensional thing that we might draw on a page. It's a big three-dimensional molecule. So we've got this long polypeptide chain. And then let's imagine that we've got a few hydrophobic amino acids, so a few amino acids with some non-polar side groups, and they're quite close together. So I've given some examples here, alanine, valine, leucine, but you absolutely don't need to worry about those. You don't need to know the names of any examples of amino acids that have any particular properties. These are attracted to each other. They're drawn together and they form clusters. They pull the molecule so that they are on the inside. They form a hydrophobic core. So any amino acids that are not hydrophobic will face outwards on the aqueous, towards the aqueous solution, and these ones will face inwards. Imagine how that would pull the whole molecule and start to create shape. So these hydrophobic interactions are really, really important in the folding of the molecule to form the tertiary structure, which is hopefully why you can start to see, thinking backwards, why the sequence of amino acids is important in creating shape. Because if we place lots of hydrophobic amino acids near each other, they will form these internal hydrophobic cores, and we will get this shape formation. In addition, if you have two cysteine amino acids close to each other, the sulfur atom in their R group, in their variable region, will form a disulfide bridge. Disulfide bridges are an example of a covalent bond. These are actually the weakest bonds in the tertiary structure of an amino acid, even though they're still actually quite strong. But the disulfide bridges are usually the weakest link in any tertiary structure. And then we have got the ionic bond. So the ionic bond is when any positive and negative R groups come into close contact with each other and they form an ionic bond. And that's basically it. We can see that in the tertiary structure, there are 4 bonds present: ionic bonds, disulfide bridges, or covalent bonds, hydrogen bonds, and hydrophobic interactions. The culmination of all these bonds creates the tertiary structure of a protein. And for many proteins, this is the end point. Many proteins are tertiary proteins, and this is the final stage of their formation. It's really worth stressing that no enzymes are required to form these bonds. They just spontaneously form due to the properties of the R groups of the different amino acids. Let's move on to look at quaternary structure. When more than one polypeptide chain is joined together by intermolecular bonds, we call this the quaternary structure. Now, this can be more than one polypeptide chain, or it can be polypeptide chains attached to a prosthetic group. A prosthetic group is a non-protein region, and the one that you'll be most familiar with is the quaternary protein hemoglobin that is made up of 4 polypeptide chains, and its non-protein group, its prosthetic group, which is the heme group that contains iron. There's just one final thing I'd like to touch on before we finish up today, and this is the difference between the 2 types of tertiary proteins. So there are 2 types of tertiary proteins. There are globular and there are fibrous proteins. Generally composed of long and narrow strands and tend to have a structural role. So they are something, like for instance collagen or tendons. Whereas globular proteins tend to have a more compact and rounded shape, and they tend to have a functional role, so they do something. For instance, an enzyme is a globular protein. It has a more complex 3-dimensional shape. Or myoglobin is a globular protein. I'm not sure whether you will have heard of myoglobin before, but it's a protein very similar to hemoglobin. Its job is to bind with and store oxygen, and we find it in our muscle cells, so it's like a final storage of oxygen in our muscle cells to prevent us having to go into anaerobic respiration when we're doing heavy exercise. Okay, so in this video we have looked at secondary, tertiary and quaternary structure. We've also looked at the types of tertiary proteins, and we've looked at the bonds that are formed within each. We've also looked at examples of each. It's really important that you learn these things, because as you go through the A-level, you're going to build on this knowledge and it's going to become important later. But that's it for now. Well done.